Alrighty. Hello, everybody. Welcome to technically the last microbiology lab. Sort of. Um, we'd be throwing a big old celebration right now if we were actually together because you guys finally made it. This is the last lab that you would have had to do. That you would have had to do. Um, besides the unknown. So let's get right on into this. Today is Lab 17, multiple test media. I just thought this meme was very appropriate for the situation that we're all in. Um, Rest in peace to our bile esculin and sodium hippurate cultures, our tubes. We never got to see the results of those because we put them in the incubator and then we never went back to campus. So I just thought this was appropriate. Hope you're laughing at home. So funny. Cody, you are a comedic genius. Anyway, let's get into the purpose of today's lab. So the purpose is to demonstrate the use of multiple test media and to compare and contrast the different biochemical tests performed that we're doing today. So just a short introduction. Um, as the name might suggest, using multiple test media has a great advantage to us as scientists. It saves us both a lot of time and a lot of money when we're trying to identify various types of bacteria because we can do one test that tests for multiple different things all at once. Um, so you can imagine why that is useful rather than just doing one test that gives you one result. So two multi-test media that are commonly used in the lab to identify enteric bacilli that we would have been doing include Kligler's iron agar, or KIA, KIA, and sulfide indole motility medium, or the SIM test, SIM, or the SIM medium. So the Kligler's iron agar, KIA test, it is a differential medium used to differentiate between members of Enterobacteriaceae, which are gram-negative enterics. We know that because we already talked about MacConkey agar and how it inhibits the growth of gram-positive bacteria. So we know that we're only dealing with gram-negative. The differentiation that the KIA test gives us is based on two factors. The first factor is the ability of the organism to use glucose and or lactose either aerobically or anaerobically for metabolism. And the second differentiation factor is the production of hydrogen sulfide, or H2S, from the breakdown of sodium thiosulfate. So these are our two differentiation factors that makes the KIA test special. The KIA medium contains 1% lactose and 0.1% glucose. It contains phenol red, which is our pH indicator. We've talked about that quite a bit. Excuse me. We've used it a little bit in the previous labs. It also contains sodium thiosulfate as a source of sulfur. It contains ferric ammonium citrate, which is our indicator of hydrogen sulfide. It also contains peptone and agar. So the KIA agar, the tube forms a butt and slant. You'll be able to tell what this looks like in the results pictures. But basically, a butt and slant means that there's a slant portion of the agar that leads to a butt portion. It's a bigger portion in the very bottom of the tube that leads up to the slant. Metabolism of sugars by facultative anaerobes may take place aerobically using the Embden-Meyerhof pathway and the tricarboxylic acid cycle, or the TCA cycle, or metabolism of sugars can occur anaerobically using the Embden-Meyerhof pathway and then various fermentation pathways from pyruvic acid. So sugars can be metabolized in one of these two ways depending on if it's aerobic or anaerobic. And under anaerobic conditions, most enteric bacteria use either the mixed acid or butanediol pathways. We've already talked about these in the last lecture. Um, if you need a refresher, feel free to go back to Lab 16. I talk a little bit more about the mixed acid fermentation pathway and also the 2,3-butanediol pathway. So just some more about the Kia agar, the Kia test. When bacteria use the mixed acid pathway, it results in the formation of H2 and CO2. We detect the presence of H2 and CO2 by looking to see if there are any gas bubbles forming, if there are any cracks or breaks in the media, or if we see the agar itself lifting off from the bottom of the tube that the test is taking place in. All of these things tell us whether or not H2 and CO2 formed. Hydrogen sulfide, which is H2S formation, is indicated when a black precipitate forms throughout the tube. So if we have hydrogen sulfide forming, we will see a black precipitate forming throughout the tube. So this next slide, we're going to talk about how to interpret reactions on the slant portion of the Kia tube. There's a slant portion that we have to read, and then there's a butt portion that we have to read. So let's first talk about the slant portion. So the glucose concentration, 0.1%, is 10 times lower or smaller than the concentration of lactose, which is 1%. So maybe not surprisingly, the glucose will be used first. There's a lot less of glucose. Organisms want to use that one up first. They prefer it. If an organism cannot use lactose as a food source, it will use up all of that glucose that's in the media, that 0.1% of glucose that's in the media, and then it'll start using the proteins in the medium as its second choice of its food source. Because lactose is present, yes, but the organism does not have the ability to break it down for a food source. So once glucose is all gone, which doesn't take very long, the organism will start using the proteins in the medium for its food source. Protein breakdown, the act of breaking down these proteins produces ammonia, and it'll turn the slant red, which means it's becoming more basic, more alkaline. And a situation where formerly acidic conditions on the slant, so it was yellow, revert back to alkaline or red, is called alkaline reversion. So if something is formerly acidic on the slant and then it starts to revert back to an alkaline environment, it starts to turn red again, that's called alkaline reversion. So if something starts using glucose, the slant may turn yellow. But then once the glucose is all used up, the organism is going to start using those proteins in the medium, and it'll turn back to red or alkaline. That's the whole idea behind alkaline reversion. So an organism that uses both glucose and lactose, if it's able to use both of these, the slant will become yellow and slightly acidic. Breaking down lactose produces lactic acid and lowers the pH. And the pH indicator in this Kia test tells us that it becomes more acidic and it turns yellow. If the organism cannot use lactose or glucose, the slant does not change or it turns red. So it would turn red if the organism started to break down those proteins in the medium. Okay? So now let's talk about interpreting the reactions in the butt of the tube, that bottom portion of the tube. But anyway, the medium in the butt is anaerobic. Excuse me, sorry. The medium in the butt is anaerobic, and the metabolism from pyruvic acid follows the mixed acid or that 2,3-butanediol pathway. If the organism can use glucose or lactose, if it uses either one of these, the butt will be yellow. meaning that the pH dropped, the organism used either glucose or lactose, and the butt turned yellow because there was that drop in pH. If the organism is not capable of producing if the organism is not capable of using glucose or lactose, it can't use either one of those as a food source, we'll see no change in the butt. Some organisms produce hydrogen sulfide from sodium thiosulfate in acidic conditions. So when the environment is acidic, some organisms have the ability to produce hydrogen sulfide, or that H2S we've been talking about, from sodium thiosulfate. We use ferric ammonium citrate to detect the presence of hydrogen sulfide in our tubes, and the reaction only takes place in an environment that is acidic. So if the butt becomes entirely black, we may not be able to see that the color is yellow. So if it's acidic and we want to say, oh, what color is the butt? I can't see the color of the butt because it's black. I don't know if it's yellow. If it is black, you can say it's acidic because this black precipitate only forms when the conditions are acidic. The ferric ammonium citrate will only detect it in the acidic environment. That's like a tongue twister, sorry. So even if you can't see the butt of the tube, and it's black, you know automatically that it is acidic. So this next slide here is just showing you sodium thiosulfate gets broken down into hydrogen sulfide, which is detected by the addition of ferric ammonium citrate, and then a black precipitate will form if hydrogen sulfide is present in that acidic environment. Okay, so that was the Kia test, the Kligler's iron auger. So now let's talk about the sulfide indole motility auger, or the SIM auger, SIM. So SIM tests use hydrogen sulfide production, indole production, and motility. It uses all three of those things. It can tell you, is hydrogen sulfide produced, is indole produced, and is the bacteria motile or non-motile, meaning does it have the ability to move around. So you can see that this also is a really good multiple test media because with one test, we're testing for three separate things, right? So that's really beneficial to us as scientists. We can tell a lot just by one test. The medium for the SIMmed auger is semi-solid, and the way we inoculate this semi-solid media is by stabbing the media 2/3 of the way down in a test tube with a needle, and then once we're in the media a little bit and we're 2/3 of the way down, we pull that needle straight back. We don't want to go to the side at all. We want to go right back where we came from. And the semi-solid media contains less agar than normal agar. Normal agar contains about 15 grams per liter of agar, whereas this SIM agar contains 3.5 grams per liter, which makes it semi-solid. And why is being semi-solid important? Well, one of the things that the SIM auger tests for is motility. So because this is a semi-solid auger, it allows for us to observe the growth of both non-motile bacteria along the inoculation stab only. So if we see bacteria start to grow along our where we stabbed the media, that means that the bacteria is non-motile. It did not move at all. It just grew where it was inoculated. However, because it's semi-solid, the bacteria can move around in this auger, and motility is detected by observing bacteria that move throughout the tube. And what this looks like, if bacteria are motile and they have the ability to grow outside of our inoculated stab location, they'll start growing all throughout the tube, and it creates a cloudy appearance in the tube itself. And just like with the Kia test, ferric ammonium citrate is used to detect hydrogen sulfide, if it is present or not. So just some more on SIM auger. We mentioned that it does three different things: hydrogen sulfide, indole, motility. So indole testing is done in the same manner as with tryptophan broth that we talked about in lab 16. We add Kovacs reagent to the broth. We get a formation of a red colored ring in the test tube if it is positive. The SIM test, however, the SIM auger, is a lot more sensitive than the Kia test for the detection of hydrogen sulfide. The SIM media lacks carbohydrates like glucose and lactose, which suppresses the production of H2S. So because the SIM does not have these carbohydrates, it suppresses, the media lacks carbohydrates, which suppresses the production, okay? Alrighty, so let's talk a little bit about results now. We can't really go over what you guys would have been doing for methods, but if you wanted to read that, you could. The most important part really was the SIM auger with the needle, stabbing the needle into the auger and then drawing it right back out because that helps you test for motility. So let's go over the results a little bit. Here is the first picture of our Kia slant. So left to right, we see on the left, the very far left, we have an uninoculated Kia slant. It's totally red all throughout. It's not inoculated. That's what it looks like before anything happens. Okay. The second one from the left is, you can label that one as K over K. And the way that you label these tubes is you look at the slant first and you say, is it red or is it yellow? Red is alkaline, or K. Yellow is acidic, or A. So what you do to label these tubes is you look at the slant. That's what the first K is in that second tube. So it's second is K divided by K. So the first K is the slant. It's acidic. It's red. I'm so sorry. It's not acidic. It's alkaline. It's red. It's alkaline. And then the butt portion of that tube, where it's flat on the bottom, is also red, which means it's alkaline. So you can label that tube as K over K. And this one's kind of hard to tell, but if you look closely, you can tell that the slant is a little bit more of a deeper red than the uninoculated. So compare the slant from tube 1 to the slant on tube 2. You'll see that tube 2 is a little bit darker on the slant, but the butt portion is the same. Okay? And then this third tube here from the left, you'll see that the slant is alkaline. It's still red. And that the butt is acidic because the butt turned yellow while the slant remained red. So the slant is alkaline, the butt is acidic, so we would label that K over A. And then this last tube, the whole tube turned yellow, so that would be acidic all throughout, so A over A. And then if you look really closely in that last tube, the one on the right, you'll see that the auger itself actually came up from the bottom of the tube. So that is an indication that gas was formed, that H2 gas and the CO2, okay? So that's what it means by auger lifting off the glass test tube, or the auger breaks or cracks or bubbles form. That is just an indicator that gas was formed. That auger completely rose above the bottom of the test tube. So here's another picture, just a different variation of what a Kia slant might look like, the results. So both tubes you would read as K over A because the slant itself is red, therefore it's alkaline, it's K. And then in this first tube especially, you can tell that the butt is yellow. However, in the second tube, you can't see the color of the butt, right? It's all just black. But like we said, that black precipitate only forms in an environment with that's highly acidic. So we can assume, even though we can't see the yellow color on the bottom of that second test tube, we can assume that whole environment is acidic. And that's why both tubes would read as K over A. Okay? So K over A. And some species will fill the entire butt with ferric, that black precipitate, but others don't. So this is just a variation that we wanted you guys to be able to look at. And then the last slide here for the Kia tests. These are just from an online, someone did this test and then they posted the results online. Test tube A here, you can see it's acidic all throughout. It's yellow throughout the entire tube. So you would label the slant acidic and then the butt acidic. And then you can see that the media is broken up and it actually rose from the test tube. So you know that there's gas there. And compare that one to B. The tube is also acidic all throughout. It has an acidic slant. It has an acidic butt, but there's no gas formation, so it looks like that auger stayed in place. Tube C, you can tell that the slant is alkaline, or K. Um, it's because it's red, and then the butt is acidic. Test tube D, is alkaline or basic at the top on the slant because it's red, and then it's acidic in the butt because it turned black. You can't see that yellow, but you know that the black precipitate only forms when that environment is acidic. So you would say it's K over A, and then you had the production of hydrogen sulfide. Okay? And then the last test tube here would just be K over K. It looks a lot like an uninoculated Kia test tube. However, they did inoculate it, and they found that it was just basic throughout the entire tube. So now let's talk about the SIM test. The results read as sequence of plus or minus, and they read in this sequence, so sulfide, indole, motility. So the results look like plus, plus, plus. That means it's positive for sulfide, it's positive for indole, and it's positive for motility. Okay? So let's just kind of go through these. This first one here is positive for sulfide. It's negative for indole because we didn't have a red ring forming at the top there. And then motility was also positive. Sometimes it's hard to see, like this first tube right here. We said that it was a positive motility result, but it's really hard to see, right? Because your sulfide result was positive. And if the sulfide result is positive, sometimes it'll obstruct your view of if the bacteria was motile or not. And that's okay. Sometimes you just have to say it was too black to read it. Like I couldn't read it. So let's go over the second tube. It was positive for all three, which means it was positive for sulfide. That's why we see the black precipitate. It was positive for indole, which is why we see our red ring up at the top of the tube. And then it was positive for motility. Again, this one was kind of too dark to see if it was motile, but you can kind of look in there and see it, and there's some other ways to tell. But we're not actually doing the test, so that's not super important for you guys because you haven't really, you're not able to look at an actual result. But let's look at the third tube here. It is negative for sulfide. That's pretty clear, right? There is no black forming in this test tube. It was positive for indole. That's also pretty clear. We have this nice red band, that pinkish red band up at the top of our media. And then you can very clearly see that it is not motile. You can see where this person stabbed into the media. This is the line of the needle all the way down here. And it's not cloudy in there, which means the bacteria was not motile. It had no ability to travel around that test tube. And then tube 4 here, it was negative for all of them. We see no black, so no sulfide production. We see no pinkish red band, so there's no indole negative. And then the growth stayed within where the person inoculated it with their needle, so negative for the motility. However, the fifth tube here, it is not black, so it was negative for sulfide. We don't see any red ring really, not like in tube 3. So that was negative for indole. However, you can really see the difference between test tube 4 and test tube 5, how they look different. Test tube 5 is a lot more cloudy than test tube 4, and that's because the bacteria in test tube 5 was more motile than in test tube 4. It could get out of that inoculation needle spot and then grow throughout. Okay? And then this right here is just your table on page 74. I'm going to give you a second just to pause this because it would take me forever to go through and say what all this is, but you guys know what it means, right? We talked about the Kia test. If it's acidic or basic, and is there gas produced? Is the black precipitate forming? That means there's hydrogen sulfide. And then those are just the results, the sulfide, indole, and motility. So I'll let you pause it right here, write these down, and then there's a couple notes I want to go over about this. So pause it right now. Okay, welcome back. I hope you had a beautiful pause time. This feels so weird. I'm just like sitting in my room talking to myself, and I just told you to pause it, and I just waited here for like 3 seconds. It's pretty strange. Anyway, um, just some notes on page 74. Enterobacter aerogenes and Klebsiella oxytoca exhibit alkaline reversion. So just jot that down. Enterobacter aerogenes and Klebsiella oxytosa exhibit alkaline reversion. Go back, if you can't remember what that is, go back and remind yourself. It's earlier in this lecture what alkaline reversion means. And then in your table on page 74, from Proteus vulgaris down to Morganella morganii, just write a note that these are not lactose fermenters. They cannot ferment lactose. That's just important to know. And sometimes the motility test cannot be read because the tube is too black. We saw that with our results, right? We said that the sulfide was positive. However, because the sulfide was positive, it was too hard to read if the bacteria was motile or not, because you can't really see if the test tube is cloudy if the whole thing is black. So two test tubes specifically on page 74 were too black to read, but we just call them positive, and that was Citrobacter frundii and Proteus vulgaris. So the motility results for Citrobacter frundii and Proteus vulgaris are both positive. However, it was kind of too black to tell. We'll just assume they're motile. Alrighty, that's all I have for you guys. This is the last lab lecture I'll give. It's been great. I really miss you guys a lot. I hope you're all doing well in your respective quarantines. I hope nobody's feeling sick or anything. Like I said, if you guys have any questions about anything, please feel free to reach out to me. You have my email. I even gave you my phone number, so you feel free to call me or text me. I want to be as available as I can because I know it's difficult to learn online. So if you have any questions, I want to be here for you. Also, if you're still listening, if you made it this far, good on you. Just one more thing to say, and it's about the unknowns. Probably tomorrow, which is Friday the 20th, or Saturday, March 21st, I'll be posting one more video just describing what I want to do for the unknown. This video is kind of long, so I don't want to get into it at the very end of this video. I'll just give you guys a break for this video, and I'll just describe it later. So keep an eye out for that. I'll send out an email. I'm going to describe how you have to do the unknown and how we're going to go about it, and that'll be its own separate video because it might be like 10 to 15 minutes of explaining. And this is already a half an hour long. I don't want to make it any longer. Okay, I will talk to you guys later. Much love. Let me know if you have any questions. Peace.